the main goal and aim of my lab is to combine and to explore and study classical problems of neurobiology like phototransduction, ionic channels, and motility with classical tools like electrophysiology, and we combine it with the most advanced methods from nanotechnology.
I'm a PhD student here. My name is Wasim Sayyed. I came from India. Here, using this optical tweezer as a tool to measure the forces generated by the neurons. A neuron is there, and in front of this is called a growth cone, in which you have the lamellipodia. The lamellipodia is now hitting the bead. With the help of these traces, one can find out what is the displacement of this particle from, let's say, from here to here. During these experiments, we have seen that this lamellipodia, they protrude and retract. So here is the new type of experiments we started to do. We started to acquire the video, the Z-stack imaging of the whole neurons, and about which my colleague will tell you more.
So I'm Hiba Shahatli. I'm originally from Lebanon, but I did my PhD in the US. Using the same setup, we are able to acquire long-time videos of the developing neuron, so we can transform these bright field images into a real-time 3D data of the cell. And the next step would be to develop mathematical models to try to understand the inside machinery that's acting to generate such a complex behavior. A great challenge for modern medicine is to develop successful neuroregenerative techniques for spinal cord injuries or other nervous system disorders or diseases. And so we're trying to find a very small piece of the big puzzle.
My name is Monica Mazzolini and I work in the Professor Torre group. Working with the retina and in particular with the rods, I needed of a completely dark condition, only with a small red light. And I use two different kinds of technique. So I combine the old electrophysiology, named suction electrode technique, with new devices used to stimulate the cell. Go! We have to draw inside the glass pipette the inner segment of the cell. After this procedure, we have to move the cell near the optical fiber. Then we stimulate first of all the rod with diffuse light, and we observe this kind of response. And then we use the optical fiber to stimulate in a local way with a general stimulation of the cell, so with a diffuse light, we stimulate basically the entire cell. With a localized stimulus, we can observe the response in a specific region of the cell, and so we can study the diffusion of the molecule inside the external segment.
The main message of my lab and the main theme is interdisciplinary. So we combine standard tools, classical electrophysiology, with modern development of nanotechnology.